enough. Because oftentimes when protein you carry out a task, 20 amino acids is not sufficient. In this case, the protein will recruit other chemical to it. So oftentimes these other chemicals can come into the form of ions. In this case, it's iron or other metal like zinc or and so on and so forth. Or they can carry meaning organic molecules and like vitamins and those things. Those non-amino acid protein, non-amino acid chemicals can be added on top of the 20 amino acids to further broaden the kind of biological functions that a protein can carry. In this case, a good example is obviously the heme group where iron is coordinated by a bunch of organic side chains, and that is embedded in the protein. Help us to breathe or help our muscles to get oxygen. So, but back to the main point that we want to emphasize is that if there is anything we can generalize about tertiary structure folding, really for the most of soluble protein in the cellular environment, one outstanding feature is they have these organizations in a way such that all the hydrophobic residue form inside the protein, and the hydrophilic residue or ionic residue are formed outside. So this, like I said, it's hard to really talk about many different proteins in general, but if anything, this is very general. So this comes to the reason that the main driving force for protein fold properly in an aqueous water environment is because the hydrophobic residue like to clump each other. Very much like when you pour the oil droplet in the water, the oil droplet will start to cluster together. It's the same principle, the same driving force. And also, we mentioned that this is due to the water release and entropy effect. So because of that, you can actually take this myoglobin structure and take a slice in the middle. So this is actually, if you look at normal outside, you can see mostly this blue color, which is hydrophilic and ionic residue. And then the yellowish color are, you can keep them to see a little bit on the outside. But if you actually take a slice cut in the middle of the protein, and then you can start see the core of it. And the large yellow color residue are hydrophobic. So this is a very commonly seen feature of almost any soluble protein. Because of that, actually, I posed the question last time. You can see this type of feature by when protein folding, you can look at the beta sandwich. I think we mentioned that if the beta strand is on the beta sandwich on the one face of it, you can actually see the residue at the sequence level. You can see the residue alternating between hydrophobic. Let's say this is the inside of the protein, hydrophilic, hydrophobic, hydrophilic, hydrophobic. So if you actually notice an amino acid sequence doing this, you have a very good chance to guess that's actually a beta strand, and that beta strand is actually seen on one face of the beta sandwich. And you can even predict which face is formed outside, which is formed inside. And then with that knowledge, you can sometimes make a prediction about why a mutation can cause strong disease. Because if you notice a gene of your favorite topic, if you have a mutation from hydrophobic to hydrophilic, when it's formed inside, and then you can say, oh, the protein becomes unstable because now your hydrophobic core is now embedded with hydrophilic. So those knowledge are useful because you can use that to make a lot of scientific hypothesis before you even solve the structure. So I actually posed a question for the class to answer. That is, if I have a protein that folds like this, oftentimes we refer to this phenomenon as an antipassive structural feature. It means the protein has one phase, or the secondary strand has one phase that's hydrophilic, the other phase is hydrophobic. So let's just say this case line right in front of us, one phase from the point inside, interior, which would be hydrophobic. So now I'm asking you, by knowing the structural feature of the here, what would you expect every how many residue you can see a hydrophobic residue? Has anyone thought about this? Anybody thought, how about we take a while to look at this? So the way to think about it is you can either draw a straight line down here, say, oh, I wanted this to be hydrophobic. So if you're counting the residue, so that's hydrophobic, right? So then, so let's say this one, draw a straight line. The tricky part, unlike our beta scale, which is easy, because it's straight up and down. Now here, remember, the alpha Higgs periodicity is 3.6. It's not integral. So you can't count, every 3, every 4, right? Because the 3 will be short by. So when you draw a straight line, what happens is, if this is hydrophobic, and this whole face is supposed to be hydrophobic, these two residues can both be hydrophobic. So it will be 1 hydrophobic, 3 hydrophobic, 4 hydrophobic, 7, this is 8 hydrophobic. So the real periodicity of the alpha helix is actually every 7 residues. So it's only, so basically it's 1, 3, 4, then goes to, then 1 plus 7. So this is how you go about it. You pretty much can draw the line. So I can, what I'm trying to do is, for example, if I draw out a sequence and tell you this is an alpha helix, I wanted you to tell me whether the alpha helix is anti-empathic or not, and why you should use this helical structure to predict. You can say, yeah, I'm counting here because I know 1 is hydrophobic, 3 is hydrophobic, 4 is hydrophobic, go to 7 or 8 is hydrophobic, and then you can pretty much say, yeah, this is my rationale that this helix is anti-empathic. So that's what I want you to be able to do. And it's actually a knowledge that's very useful, because whenever you study a protein, and the first thing you get is the sequence by chromosome, and from the sequence you can actually derive a lot of rich information like this, and that can be very useful to guide your thinking. Okay, so that's basically related to the tertiary structure folding to our discussion of secondary structure. So these are all interrelated, and then they can be very helpful skill to in terms of analyzing protein structure and predict function. Yes. So I think this should be 8 residue. So every 7 residue. So starting from here, if I consider this is a hydrophobic part, now it's randomly called. So hydrophobic means this guy's hydrophobic, right? So we call them hydropathic. So and then this is facing on the other side. Second residue and third residue is on the other side of the helix. So they should most likely be hydrophilic. Now the third residue, and I'm counting 1, 2, 3. So the third residue should be starting not pointing the same way as this one. So that should be hydrophobic. The reason the fourth reason is mostly hydrophobic is because remember the alpha helical periodicity does not set on the 3, 4, 4. It's actually in the middle. So because of that, 3 and 4 are often also hydrophobic. And then you go 5 should be hydrophilic, and 6 is also hydrophilic. This is another very good way to see it, but it's actually pointed the other way. And then 7 now it's going back. 7 is the periodical going back to a full 2-fold, right? So 3.5 times 2. So this guy is in phase with this guy. So this residue, this residue, this residue, this residue should be hydrophobic. And then their spatial relationship is what I just told you. You're counting from counting from here is 1, 2, 3 would be hydrophobic. This 2 would be 4 and 5 would be hydrophobic. 6, 7 would be hydrophobic, and 8 one would be. It depends on how you're counting. So if you're counting from here, then the number will all reduce by 1. But it doesn't really matter. The key is you should be able to line all the sequence mentally on the structure and then make a prediction. Right? So I'm not going to tell you, oh, it's every 3 to 4 residue. I'm going to just give you the sequence, ask you to make a prediction. I'll tell you that the alpha helix, and then you should be able to apply the knowledge to pick up the residue, the regularity, and tell me that alpha helix is anti-passive or not. Any more questions? This is pretty useful. Yes.
So you say it has to be hydrophobic every 7 amino acid, but I count 3.
Not just every 7. The 3 and 4 will also be.
Oh.
So because you can't be hydrophobic and a bunch of hydrophilic, then you can't be, because we're talking about this guy, the whole thing is packed on the interior. So so you can think of on the back of the helix, it's all hydrophobic. So you can think of this, you can take this whole helix, just try to put it over here. And I want this guy all be hydrophobic. And then you should be able to tell me where these residues positioned on the primary sequence should be, how the predicted it should look like. So this is a useful thing that you can do when you analyze the structure. Well, from the sequence of get structural information. Any questions from this one? Okay. But that's it. We're talking about soluble protein in intracellular, but oftentimes you also find protein in the transmembrane region. So these proteins, they're not sitting in aqueous solution. So you already know the folding principle for this class protein will be different. Indeed, for membrane protein, when they embedded in the hydrophobic environment, this protein has the opposite folding pattern. That is, they on the outside of the protein, which is based the hydrophobic lipid environment, and they have hydrophobic residue. Oftentimes, these proteins are involved in transporting nutrients, sugar, ions, so they have a pore channel. These pores are dealing with hydrophilic chemicals, so therefore, their so-called inside or their pore, this is actually an aquaporin that transporting water molecules. So there obviously the little pore region will be hydrophilic. So so when we talk about the folding principle or hydrophobic driving force, we need to talk about the context. For intracellular aqueous environment protein, the driving force is hydrophobic interaction. The inside of the protein is most hydrophobic. For membrane protein, it is the inverse. The outside of the protein are most hydrophobic, where the core region are largely hydrophilic. I mean, we can go on talk about many structures, but other than the principle that I just trying to help you to apply some of the knowledge of the hydrophobic interaction, also on the secondary structure of optic base, that you can make some prediction on what the protein sequence might look like in terms of protein structure. For the majority of the time, the protein structure need to be experimentally determined before you do anything. But also for the analysis of the protein structure, oftentimes can be very tedious and also somewhat difficult to unify because every protein structure has its own features and shapes and therefore explains its own unique function. However, after solving many structures, we have discovered that nature don't just make every new protein new. It appears to be that they use some, in addition to the very basic alpha helix and beta strand, sometimes they put certain alpha helix together in a very fixed way. And this is what we sometimes call supersecondary structure, or we call them motif. Meaning, you see a helix kind of linked by loop, and then docked to another helix by a certain angle. And you can notice this kind of feature in many different proteins. So apparently, nature has created a set of folding pattern above the just simple alpha helix and beta strand, and they use these simple folding patterns many times to make up different proteins. So this type of a motif feature really helps you to analyze protein structure between the very basic alpha helix to this very complex folding pattern. So right now, structural biologists try to collect a lot of this motif. So when they analyze a very complex protein structure, they will go looking for their familiar motif. So if you have to go to a database, you find a new sequence, there's an ability for the computation now to predict what kind of motif might be present in a protein you just cloned. So that's one thing that in the protein characterization between when we talk about tertiary structure, we use the concept of motif a lot. So in a way, motif is something beyond the alpha helix beta strand, very basic secondary structure, but a set of secondary structure that form together in a fixed way and that present in many proteins. For example, just one example that we can see in many DNA binding proteins, they use a motif, or we call helix loop. Helix motif. So in this case, it's an alpha helix, make a little loop, and then turn around, make another alpha helix. So because of this finding, enough different proteins, people assign them as a commonly frequently occurred supersecondary structure, so they gave it a name called helix loop T. And sometimes they even group different proteins by this way. So this the purpose of motif is twofold. One is helping us to analyze different proteins in terms of what common structure. The other one is it helps us to guess what a new protein might be doing. Let's say, for example, if you find a protein that has the so-called base helix group, helix domain that binds DNA. Let's say you in your gene analysis and find a new gene. You had no idea of what that protein is, but you discovered that from the sequence level it has the basic helix loop protein. You can take a reasonable hypothesis that this protein might be binding DNA and do something about DNA. That's how it's functioning. So it's kind of goes both ways. To help you analyze protein structure to go down to the feature. The other one is to help you to deduce function by finding out whether that protein contains a certain kind of motif. So these type of motif thinking really is dominating the structural analysis. So we now start accumulating a lot of idea about different kinds of motifs, and this motif help us to classify proteins structurally and also functionally. So another very good example is the so-called immunoglobulin motif or domain. These are made largely beta strands, and then they can recur in many different tandem forms in the protein, and largely occur on the protein surface. So for example, this particular protein called cellular domain 4, there's 4 such individually motifs linked together. So these are the ones that mostly expressed on the surface of the T cell. Well, when HIV infects, and the HIV virus will grab on this CD4 protein and try to attach to the T cell before they make the entrance to the infection. So these type of immunoglobulin proteins can be found not only in the cell surface and also in antibodies, many other proteins. So again, whenever you see a IG fold, you immediately think, oh yeah, my protein may be either serve as a function on the cell surface as a protein attachment, but it also maybe my protein could be part of antibody or some sort of immune-related system. So that's the domain. The other usefulness of having domain concept is helping you think about protein evolution. Because you can actually sometimes see complex molecules found in higher organisms like a mammal. They're not just new proteins. They end up just tagging, like play a Lego game. They use different motifs from various proteins that of the lower organism. For example, this mammalian phospholipid looks like a very complex protein, but if you have applied the domain concept, you'll discover that it's just a simply link a bunch of sub-motifs or domains found in various different proteins, and they just put them together, make it a multifunctional protein. And this actually happens a lot. So again, structurally classify proteins by motif help us to decompose a very complex protein into manageable, well-defined domains for motifs. And also help us to understand there might be some evolutionary linkage between complex genes in higher organisms and how the lower organisms evolve. So that's another usefulness of analyzing protein in terms of well-defined motif and domain. So up to this point, we're talking about how polypeptides go from a helix base strand forming a tertiary structure and how the tertiary structure in terms of the motif can help understand protein. But there are even higher order structures. In this case, we mentioned very briefly before, that is, two different polypeptide chains or two tertiary, individually folded tertiary proteins can come together in many different ways. They can come as a dimer if they're same unit and homodimer. There can also be a heterodimer when they're different subunits. And go back to the oxygen carrying protein. In addition to the myoglobin and the hemoglobin found in the blood, also is a very good example where not only you have individual peptide, both a crystal structure, you can have 4 subunits come together and form this very very beautiful high order structure, has some kind of the twofold diet symmetry. And the reason for this is multiple, and sometimes protein form a multiple molecular structure is trying to build up a particular mechanism for function. For example, in this particular case, hemoglobin, nature tried to create a enhanced response to the oxygen level. So they want to have a cooperativity built in the sensory system. What I mean by that is, for example, you want to say, well, if each of these subunits is responsible to carry oxygen, then you want to say, what's the point of building a full subunit cooperative structure? Well, nature has evolved such that if this particular subunit binds oxygen, it will induce a structural change on it. If there are other subunits that physically bond to each other, this change can convey to the next one, such that the affinity for the next subunit to oxygen can be enhanced. So therefore, the binding for oxygen is not going to be a very shallow response, but rather very sharp cooperative response. Meaning the very first oxygen binding will make the next oxygen binding much easier. So this is actually just one example of how high order cooperative structure can be evolved in nature for purpose of building up cooperativity. Meaning that subunit communication can tell, hey, I want this activity and I want more of this activity. So when this activity happens to one subunit, it has a physical way to communicate to the next one through this type of high order cooperative structure. And also there are other similar examples, not limited to the cooperative structure of attachment. Remember I showed you the example of the nicotinic receptor, that's a pentamer. And you also have proteins that assemble as a hexamer and even higher order structure. And the most prominent one is the virus. The virus usually have a multiple cells assembled as a shell. And they have this so-called icosahedral symmetry. It's like a little, very beautiful assembled. They usually have a mold. They don't have that many different conjugating, but they use the same conjugate to assemble a very sophisticated shell. And this is the rhinovirus, which is the virus called the common flu. I don't believe it's the one that going on right now. I've heard that the one that going on right now is called H1N1, which is the common virus. But anyway, so this is just the ultimate example of how protein can build up sophisticated structures going from hierarchically from every different level all the way up to hundreds of units like this. So ultimately, as a structural community, of course we can solve many structures. But one fundamental question that has driving the thinking is how does a particular protein fall in a certain structure? And then, or another way to ask the question is, what determines the structure? Is it the sequence of the protein itself, or maybe something else? Maybe when the protein is produced in a particular cell, there's some sort of template to mold the protein into certain shape. So this is actually a non-trivial question because it fundamentally affects our understanding of nature. So that's why this experiment, even though it's done in 1950, is very fundamentally impact our understanding of protein sequence into structure relationship. So it's actually worth talking about a little bit here. So this is working done by Christian Hankinson on a protein called ribonuclease, which is an enzyme in various activities. So he had a very simple experiment to test the idea that whether the sequence of the protein itself has the intrinsic ability to guide the folding of the 3D structure, where you don't need invoke any template mechanism that we still don't know anything about it. So the experiment is pretty simple. You start with a protein that's active, isolated from nature. You can say it's made by some mysterious mechanism in terms of its structure. So you can destroy the protein by a number of methods. So remember, we talked about forces hold protein structure. The force including hydrogen bonding, hydrophobic interaction. Also, we talked about sometimes with disulfide. So that's why when you try to break up a protein, you want to break up at least some of these interactions. And so oftentimes urea, which we'll talk a little bit very briefly, is the one that can break up a lot of hydrogen bonding and also suck up the water so you won't hydrate the protein very well. BNE stands for beta-metaclassinol, which is shown here. It has the salt hydro group. It has the ability to break up disulfide. So virtually, if you can break up the covalent disulfide and also the forces to form protein structure, you can take a native protein and make it unfold it as a nascent peptide. But the key here is that the nascent peptide, when it's not active in terms of enzyme activity, so that at least confirms the one point. The 3D structured proteins require function. So no big deal, we already know that. But the next set of experiments is the critical one. You can actually stylize away these denaturing reagents. And then let the little oxygen to oxidize these disulfides. And amazingly, in the test tube, without any help of any cellular machinery, the protein can go back to the 3D structure and reassume its enzymatic activity. So this is the experiment to set up the current paradigm, that is sequence and determine the structure, therefore its function. So this is the experiment being talked about in textbooks for years, and for that matter, it really is worth mentioning. At least conceptually for the majority of the protein, this concept is applicable, meaning sequencing indeed has all the information that needed to guide the 3D formation. And you don't need to imagine any mysterious template or molding in the cell to mold a particular protein in a particular shape. So again, we review a little bit about when we talk about protein forces that stabilize protein. So that corresponds to the reagent you use to denature the protein. For example, urea, just look at it. It looks very much like a amide bond, right? Remember, we talked about the peptide bond had a carbon atom going out, had NH come down. Those hydrogen bonding donors, etc., are the ones that are driving a lot of alpha beta formation, beta spin. This urea can be a very powerful competitor for hydrogen bonding donor receptor. This guanidine hydrochloride equally well, and on top of that, you can even break out the ionic interaction with the sulfate charge. So guanidine hydrochloride and urea are presumably by just disrupt all hydrogen bonding interaction and including those solvation effects of water, because they also form very strong interactions with water as well. So what we are doing is basically disrupt all the protein internal hydrogen bonding. They also can suck out all the water molecules in the wall. So the protein cannot form hydrogen bond itself. It cannot be stabilized by the water. And I told you the beta-metaclassinol has the sulfide group that can compete with the disulfide. So they will disrupt the system. So each of the chemical, their ability to denature protein corresponding to the forces we mentioned to stabilize protein. And one more thing that can add into this discussion is, remember hydrophobic interactions. How do we deal with hydrophobic interactions? Well, very much like you wash your greasy dish using soap. And we use a molecule called SDS, sodium potassium sulfate. This molecule has this large non-polar chain. This is the guy that can basically invade the protein interior interacting with all the hydrophobic residue. Basically, it blocks the hydrophobic core of the protein, and then drag them into the water, all water to these ionic cascade. So that's very nice. So those are the ones that, so the denaturing chemicals reflect the force you mentioned, our protein folding. So, these experiments of sequence, yes,
Did you mention what guanidinium chloride did?
guanidine hydrochloride is similar to the urea. They break up hydrogen bonding of the main chain. They also break up ionic interactions because guanidine hydrochloride has a charge. So they're even more powerful. So if you have a protein you could not dissolve the urea, try guanidine hydrochloride too. And then they usually can do better job. Okay, because of the experiment I mentioned to you that established the concept that sequence itself contains all the information to dictate the 3D folding. It does stimulate a lot of intellectual pursuit as to who will be the first person ever achieve the ability by looking at the sequence to tell you exactly how the atom is going to be placed in each individual position. So meaning you can predict the 3D structure from the you just look at the sequence. This has been the dream for scientists for the past 50, 100 or maybe at least 50, 60 years. We haven't done that, but we have done a few things trying to. So the most naive and the simple way to think about it is look at see if each residue has a preference to go into alphabetic, beta strand, or turn. You see, for each residue, there's some preference, but it's not strong enough for you to say, if I look at the sequence, I can compute, this structure is going to look like this. So there are some ability to predict whether a segment of sequence will form alphabetic or beta strand, but there's these tables, which is computed by look at all the solved structure and see each residue, what is the chance for them to be found in alphabetic, what's the chance in the beta strand. So this is the table. Obviously, this table can continue to change between the more structure we solve. But if we sample enough, we notice that this statistical distribution is about right. And there are some bias, not enough to predict 3D structure from just look at sequence. The other complexity comes from the fact that you just look at the exact same structure sequence, but if you look at in different proteins, they form different segment structures. So you can look at the 4 or 5 residual stretch, identical order. In this protein, it's part of beta. In this protein, it's part of beta strand. So that means context also dictates whether a short segment of amino acid will form what kind of structure. So this basically continue to be a challenge. And then in fact, my personal belief is somewhere down the road, maybe your kids or maybe your grandkids will come up with a way to do this. And this way would be like, depend on the further improvement of computational technology, because to calculate many different ways for the atom to put in space is non-trivial, even with current computational technology. The other way is our understanding the molecular force that dictate the folding of the protein need to be further complete. With these two, maybe one of those days. I don't know. So that's the next level of human evolution. And we can get to even better and better, further away from monkeys. That is how you can do it if you can do it. But right now we couldn't do that. So what we could do a little bit nowadays is we can use many solved structures and then by compare whether your sequence look like it's a solved structure and then use one we call homology modeling. But as an issue, by just purely look at sequence, predict the structure is not possible yet. And we're making progress, but we haven't gone there. Oh, by the way, I only know this is actually, if you want to find the most intellectual challenging problem if you're into computation, this would be one of the kind of things you can pursue. I'm pretty sure, well, a good example, analogy of this is like in 1970s when computer was just introduced, my colleague, Ariel Washoe, he thinks, well, this would be good, right? Because whenever we talk about chemical reaction, we know what's the beginning, what's the end. We have no idea what's in the middle. Because you can't experimentally observe it. So he basically devoted his whole life trying to use computation to calculate how atoms bounce each other and then reorganize, become a new set of structure. And for that, he worked on for 50 years. Now it becomes a common way to study chemistry. So for that, he was awarded the Nobel Prize this year, and you won't know that. So you could do the same. You have to come up with a way to solve this question. You know, is how can you go from sequencing into an actual 3D structure in computation? But anyway, so there are many, many challenges coming along the way. I told you not only an individual peptide can be context dependent, even for the entire protein sometimes. So this is exception to the rule. Once in a while, you'll see a few proteins in the different biological context. Although they have identical amino acid sequence, they could adopt into different structures. And you might say, how do you describe this? Well, their energy difference is very similar, so they can be altered simply by what they're interacting, how they bind ligands, and then they can adopt into very different structures. So this can further complicate our ability to predict the 3D structure purely from sequence. But the majority of proteins still follow the rule I was mentioning earlier on the ribonuclease. Most of the proteins, the sequence dictate into the coding of one unique 3D structure. So finally, we can talk a few things about. So that's just a thinking process, how to go from sequence to 3D structure. But let's do some experiments and what is the protein folding entails in terms of the intermediate state. But when we try to heat up for protein or dump urea or quantity into protein, we're observing the protein becomes denatured. Generally speaking, it's not like a protein goes through many kind of partial denatured and final denatured. They appear to be highly corruptive. What we mean by that is we don't see these little mini big stack states. Rather, either you have a native and you all have denatured. So even when you look at the middle, you have half either fully folded or unfolded. So this is from the very simple chemical point of view. These are very it's called two-state hypothesis. But in reality, the actual folding process goes through a lot of intermediate states. And then there's actually this famous hypothesis called Leveson paradox. If you take a random protein, you let each residue to take all the possible conformations, and then there's just astronomical number of protein structures you needed to go through to find the one that actually found the 3D structure. So if you actually take a computation of them, even nowadays with current computation technology, it takes a long, long time to calculate all the possible conformations. So the folding process is really not a random process, but rather what we call a energy pooling or gravitational draw. A very good analogy is that you can imagine that you blindly close your eye, there's a flat field, there's a little hole, and you try to roll the ball randomly without looking, hoping the ball will just search all the flat surface and try to drop in that hole. So the probability for that is very low, it takes a long time. But if you imagine again, instead of having a flat field, but you have a funnel, and then you can close your eye and then the hole is right in front of you, just drop it. The gravitational pull will eventually drag the ball into the hole. The folding process of protein can be pretty much described as that. So you can think of a random peptide, there are locally folded substructures, and the substrate formation basically pull the protein towards the correct structure. So it's like a gravitational pull, it's creating pulling. So the more and more the correct local structure forms, and they will start form on top of each other. When the final form is dropped in the hole. So it's very much described in this picture here. So it's pretty much like the folding process, like a pulling. So it's actually, the reason that it can happen in a reasonable faster time scale is because of it's not a total flat random search, but it's driven by creating lower fringes. For every correct folding student, they go on to the next correct folding form. And we also mentioned this. I just want to put this picture here to remind you again. For example, misfolding protein can cause disease. We mentioned all kinds of disease, but this one is an example for the so-called mitosis disease. That a soluble protein can fold into a beta amyloid, And then this can even be infectious. So if you have a protein that already start from this, they can induce the normal protein to the disease form. So this is why the famous theory that the protein can also be infectious agent, and then the principle of this is behind due to the protein folding. And this part you can actually read it. They are just general knowledge, and they're useful. And in the context of discussing protein folding, it's worth to at least remind you one more time that protein folding is not only useful to understand the basics of protein structure, but also development. Finally, wrap up is to go back to the original point. So the texture structure of the protein determines the function, but the function of protein is not all determined by its overall structure. In addition to the one that I told you that sometimes the protein function can be enriched or broadened by recruiting ions, heme, but it also can be further expanded by chemical modification of the surface of protein residue. So a few examples here: hydroxyl protein from the collagen, the carboxylated glutamate, and also the glycosylated asparagine, as well as the phosphorylation of the serine and tyrosine. Remember, this one actually was the one that I mentioned with respect to the stop kinase. When the phosphorylate is misregulated, it can cause cancer. So there can be useful for normal physiological function. It can also be involved in pathological disease. This is the most dramatic example. Not only the side chain, the main chain can even be modified rearranged. To the GFP, I don't know if anybody you work with, but I think some of you may end up working with in the future. It's one of the most incredible technology that revolutionized the study of the cellular biology, because you can put a protein into, you fuse your favorite protein with a GFP, you can monitor the protein's travel in the cell. And for that matter, this was also awarded a Nobel Prize a few years ago. And this is probably the ultimate dramatic example of how protein structure can be expanded beyond the normal folding. Because when we talk about folding, we basically assuming the peptide backbone should not change chemically, but under the certain environment, the protein backbone can undergo chemical reaction such that it will create a fluorophore that can fluorescent under certain light conditions. So I found that this one is probably a little bit difficult to do the air coaching. So the next picture I just got it from the literature. It's easy to see this because you can see the key here is you have a serine, tyrosine, glycine. The amide backbone can condense with this hydroxyl group to create a OH. That OH group will be eliminated with water to create this ring closure. So this is probably more than any regular chemical modification. And once this happens, this bond becomes highly oxidable. And that's why when you create lost water through oxidation, then you get this very extended conjugated system. That's why you start to see it harder. So because I expect most of you know the organic chemistry, this type of higher approaching should be reasonably familiar to most of you. Yes.
Is this process responsible for causing any disease, or it just happens in normal animals?
This one, this is actually a normal process. It evolved the protein, the G protein, when it's nasally produced, it's not fluorescent. So it undergo these dehydration and oxidation, then they become fluorescent. So it's not involved in disease. So it's how protein become floating and become a functional protein. So this basically wrap up our discussion of protein structure, and the next chapter will start to talk about experimental techniques that use to generate the knowledge of protein structure and function. And so that's what we'll start on the next chapter.